I want to welcome everyone who's joining us today for the NAM annual meeting, and particularly those returning from lunch, as well as those online. I'm very pleased to introduce the next panel in our program, Emerging Science and Innovation for Therapies. We've got a team that comes from various disciplines, different ways to study the brain and the mind, and it's really exciting what you're going to hear about. This panel will be moderated by Dr. Kaf Dzirasa, who is A. Eugene and Mary Washington, Presidential Distinguished Professor at Duke University. Dr. Dzirasa, I welcome you and all the panelists for this session to please join us on the stage.
Well, good afternoon. It's certainly an honor and pleasure to be here. Before we get started, I would gently ask you all to please silence your phone, at least until the start of the next panel. It's been a tremendous pleasure to be here and part of this planning committee. And as we were sort of talking about the arc of the story for today, there was some discussion amongst the planning committee. And we got into the importance of thinking about loneliness, the importance, as Josh mentioned, about thinking about rental assistance and suicide prevention and early education in schools. And there was some back and forth on the committee of whether it was important to talk about the brain or neuroscience at all, and whether there was actually a role or need, whether we should be investing our time and our resources, and what could neuroscience actually teach us about combating these problems when the social determinants of health, particularly in the area of mental health, were so clear, so present, and so important. So today we hope to explore that. And I hope by the end we will convince you that understanding neuroscience, particularly for its goal of helping us understand mental illness, is just as important of understanding checkpoint inhibitors and its integration with smoking in understanding cancer, or perhaps in understanding GLP-1s as it is exercise and diet and thinking about cardiometabolic disease, right? The brain is an organ that keeps organisms, in other words, us, alive by helping that organism function within an environment. And when I say brain, I mean brain in its supporting infrastructure, the blood, the lungs, the kidneys, but the brain helps us do that. So we hope today to explore how that organ works and how it functions within an environment to keep us healthy. And so we have a group of panelists here to explore that arc. And we'll talk about the brain, both in terms of how the organ is built and formed. We'll talk about how the brain develops and how it's influenced by environment and how it develops, both through early life and adolescence into adulthood. And then we'll also explore some really innovative new therapeutics that are coming online. And what those therapeutics and studying those therapeutics teaches about the brain and the organ. So without further ado, let me introduce my colleagues who are up here with me. Matt State, who is the Obendorf Family Distinguished Professor and Chair at the Department of Psychiatry at UCSF, University of California, San Francisco. Huda Akil, who is the Gardner Distinguished University Professor of Neuroscience and Psychiatry at the Michigan Neuroscience Institute. Damien, who is the Redleaf Endowed Director of the Masonic Institute of the Developing Brain and a professor at the Institute of Child Development at the University of Minnesota School of Medicine. John Krystal, who is the Robert L. McNeil Professor of Translational Psychiatry and Chair of the Department of Psychiatry at the Yale School of Medicine. And then Helen Mayberg, who I'll take a personal privilege. If you all look through your program, that bio is not hers. So because Helen is a fantastic scientist and a personal mentor and friend of mine, I will take the privilege of reading her whole bio so that you all have it. I'll read most of it, half of it, Helen. Reasonable compromise. Helen is a professor of neurology, neurosurgery, and psychiatry and neuroscience at the Mount Sinai School of Medicine, or the Icahn School of Medicine. She's played a fundamental role and is recognized for her role in developing neuroimaging studies of brain circuits in depression and their translation to the development of deep brain stimulation as a novel therapeutic for treatment-resistant patients. And she now leads a patient-focused transdisciplinary research team with the shared mission to advance precision surgical treatments for complex neuropsychiatric disorders. She's a member of the National Academy of Medicine, National Academy of Sciences, and National Academy of Inventors, and the American Academy of Arts and Sciences, where she participates in a wide variety of advisory and scientific activities across multiple fields of neuroscience. So with no further ado, I'll turn it over to Matt State.
Wonderful. Thanks, Kaf. See if I can go back. Delighted to be here, and I'm going to talk to you today about autism spectrum disorder and try to give you a sense about an aspirational arc from understanding the genomics of the syndrome to thinking about how we can move forward with therapeutics. So I'll start just by reminding you that the sine qua non of the diagnosis of autism are fundamental impairments in reciprocal social interaction. Currently, the estimated population prevalence in the US is about 3%. And that is a consequence, in part, of a kind of continually broadening diagnostic definition, so currently encompassing multiple formally distinct disorders. So this leads to an enormously broad phenotypic spectrum in autism that ranges from individuals who require 24-hour care, who have a dramatically shortened life expectancy, in the same diagnostic category with many individuals who do not experience the core symptoms of autism as a disability. And I would say who would object to some of the language that I'm going to be using in this talk around mutation, disorder, cure, and object to thinking about autism solely through a medical model. I'm going to address that by focusing on a subset of individuals with autism today on the severe end of the spectrum. So sort of being a diagnostic category being proposed called profound autism, but generally captures a group of individuals who have autism, who have co-occurring intellectual disability, who require 24-hour care, are minimally verbal or nonverbal, and also in that category are remarkably enriched for known genetic causes and for other severe neurodevelopmental phenotypes, including epilepsy. So what we've learned over the last 15 years or so has taught us that there is a distinctive genetic architecture for autism compared to most common adult-onset psychiatric disorders. You can see here, I hope, that it says that we've discovered about 250 genes so far through the study of rare, often spontaneous de novo mutations in the coding portion of the genome that are predicted to be damaging to the encoded protein, and where a single event in the individual is dramatically increasing risk. The vertical axis on the left, is it on the left side, is effect size, and what you can see, I hope, is that, yep, it didn't show up there, is that the top part of that graph is orders of magnitude larger than what we're used to thinking about for common complex psychiatric disorders, where typically the genetic architecture is a conspiracy of common variations in the genetic code that individually carry very small effects. So this collection of specific genes with mutations that in each individual can carry very large effects has given us a window into biology. And so right from the start of gene discovery now to, as I said, about 250 genes, we have consistent themes that these genes impact synaptic function, chromatin modification, transcriptional regulation, neurogenesis, ciliary biology. And we can ask from that list of genes not only what they do, but when and where they're having their impact. And we can do this because we can begin to intersect this growing list of genes with an increasingly high-resolution map of the molecular landscape of developing human brain. And when we put those two together, what we find is that there's an important nexus of risk in mid-fetal cortical development, and particularly in cortical glutamatergic neurons. Now, that sort of sense of resolution now can be advanced further through the study of proteomics. So I've taken you from genomics to transcriptomics, now to proteomics. This is the product of a number of labs at UCSF engaged in something called the Psychiatric Cell Mapping Initiative, where we took the 100 top autism genes and did AP mass spec in order to begin to identify the proteomic interactome of autism. And there's there's a lot here, but I'll just point out that one of the most dramatic advances that enabled this study at scale was the development of AlphaFold, which allowed us to narrow in on 30,000 potential interactions and looking at autism genes and what they're interacting with, down to be able to begin to predict with high confidence what those actual structural complexes look like. And then importantly, when we introduce mutations that lead to autism, that we can begin to predict the structural changes as well with great accuracy. So that lays the foundation to begin to think at a level of resolution where we can begin to talk about things like small molecule development, target development, et cetera, in a way that has been difficult previously. So this kind of distinctive genetic architecture has also led to the emergence now, I think, of a very important and distinctive clinical translational ecosystem within psychiatry. So in psychiatry, for most disorders, we lament the fact that we don't have biomarkers in order to be able to subset our samples. In autism, about 20 to 25% of our population is identifiable based on a known mutation in a known gene and has potentially quite severe phenotype and where there's dramatic overlap, particularly with developmental epilepsies. What that's led to over 15 years really is the consolidation of a research ecosystem, as I said, driven in great measure by family groups and advocates who have organized around the identification of these genes and collaborated with academia, with biotech, and with pharma in order to be able to advance both research and therapeutics. And so what you're beginning to see is the development of really a plausible arc, even though these are rare disorders, because of that work of these advocacy groups, being able to employ at both the research and potentially therapeutic level things like nucleic acid targeting, small molecule development based on understanding of protein interactions, and even because of the overlap with severe epilepsy, neuromodulation, which you're going to hear much more about as we get to the end of our panel. So I've taken you through sort of this aspirational arc focused on a subset of individuals with autism, and I wish that it was as linear as this first line, that we could go from our understanding of these genes to cells, to circuits, to behavior, and to treatment. But the brain and the developing brain is quite a difficult place to work, and we have to deal with a number of challenges. So one is pleiotropy. At the molecular level, a single gene can have multiple functions. We also see that kind of diversity and pleiotropy in the phenotypic outcome of a single mutation. And then we have to deal with the complexity of the kind of extraordinary dynamism and cellular and circuit level diversity of human brain, so that the sort of idealized notion that we could pull on a single gene in order to think about a therapeutic, actually, if you look at the bottom of this figure here, you pull on a gene and you very quickly end up in sort of an infinite search space and trying to traverse that path. So one of the ways, as I've described, that we've been dealing with that is to think about convergence, taking multiple genes on simultaneously and leveraging these remarkable data sets that are coming forward to be able to identify areas where at the cellular, at the circuit level, at the molecular level, that there may be opportunities for intervention. But very importantly now, what's overlaid here is really, as I said, a human clinical translational research enterprise that can go from nucleic acid therapies and small molecule development, and even because of the overlap with severe epilepsy, begin to gain traction intracranially. So what we hope is that this really is an inflection point for the study of and development of treatments for the most severe forms of autism, and then with luck, that will extend to a deeper understanding of the broad autism spectrum, bringing a new generation of treatments to those individuals who experience that as a disability and who are seeking somatic treatments. So I'll pass it on to my esteemed colleague.
Okay. So thank you for inviting me to participate in this panel, Kaf, and thank you to the NM for highlighting this topic. I'm going to switch gear and talk about affective disorders, depression, anxiety, stress-related disorders, which is, of course, a very vast disorder. I see it as a second pandemic that has expanded after the COVID-19 pandemic, and it is really global in nature, not only in the United States. And as a scientist, my question is, what can science do? I'm a neuroscientist, so I see the brain as the center of the universe, both going down in terms of genes, cells, and circuit, but of course as it affects how we feel, think as individuals, how we interact socially, and live in an environment, community, and all the way globally, with the importance of thinking both bottom up and top down. Now, in thinking about affective, and my goal today actually is to focus specifically on the idea of resilience to stress, and what scientifically we can begin to do to understand that and promote that as part of thinking about how we combat the pandemic, how we can predict susceptibility and resilience in an individual, and how we can then use potentially that knowledge to prevent maladaptive behavior. So in thinking about affective neuroscience and affective disorders, it helps to think in multiple time domains. We feel emotions in the moment. We're angry, sad, happy in this moment. There are also longer affective states that are moods, depression, sustained anxiety, mania. And then there are temperaments, which are a certain style of responding to the world in an affective way, in an emotional way, that constitute part of our personality. And it is important to think about temperament because it really kind of shapes then the kind of responses we're going to have to the world around us. And we have, over the years, created animal models for the various of these levels of analysis. One of them has been an animal model of temperament. And I'll start by asking you, are you the kind of person who would love to go skydiving? Or would you rather stay at home and be knitting? And that turns out to be this simple kind of question related to temperamental tendencies of risk-taking, the kind of behavior people engage in, and determine when things go bad, the types of disorders that people suffer through, so-called externalizing disorders, including substance abuse, internalizing disorders such as depression and anxiety. And we have created a rat model of internalizing and externalizing disorders, knitters and skydivers, using selective breeding paradigm. And we've done a lot of both genetic and neurobiological studies on them. And what is notable first is that their behavioral phenotype really maps beautifully onto the human phenotypes of temperament. And the two lessons that I'm going to give to you from these studies is A, it's very genetic, it's very heritable, it's very predictable what temperament an animal is going to have based on its parentage. And we know a lot of the genes, both at the genetic and gene expression levels, that are involved, including gene regulation, inflammation, energy regulation, and synaptic structure. But the other thing is that it's modulatable. So you can take an animal that is very susceptible to stress, very anxious, and you can change that phenotype, especially during adolescence, in the kind of environment you put it in. An enriched environment actually induces resilience, whether it's social enrichment or physical enrichment or both. So that brings us to the idea that there is more than one kind of resilience, genetic and non-genetic resilience. But even if you control for genetics, there is also the way we react to the world in a given instance and how that builds on. And for that, we've used an animal model that the Nestler lab has created of repeated social stress, where after a repetition of social stress, animals emerge as either resilient or susceptible to stress in the way that then they interact socially. And we asked, is there something in the brain the first time they encounter a social stress that carves a path to resilience or susceptibility? And to do that, we've used a technique called fast trap, which labels brain cells during that first episode, allows the animals to stay alive, undergo the repeated stress. And then we asked what circuits was active, and can it predict the future? And the answer is yes. The way the brain responds the first time actually sets the path to different neural networks that then emerge as determining resilience or susceptibility in these animals. So the combination of these animal studies told us that that it's really genes, development, living environment, and specific experiences that shape the behavior, adaptive or maladaptive, and that in animals we can predict and modulate these behaviors, knowing enough about the genetics and the biology and the brain function. So then we asked, can we take that knowledge to the human? And we did this study we called the Michigan Freshman Study at the University of Michigan, where we take young, healthy 18, 19-year-old. We know they're going to undergo stress because freshman year is stressful. And we follow them longitudinally and ask, who's going to emerge as resilient or susceptible at the end of the freshman year? We do a whole bunch of tests at the beginning of the year, and we follow them and assess depression or anxiety. They have wearables, and the question is, can we predict? And we happen to capture right before the pandemic, and then since then, every year we have a cohort during the pandemic. And as we've heard today, the rate of depression, as shown in blue, has increased very dramatically and immediately during the pandemic. But baseline depression, these kids are coming in with higher rates of depression and anxiety, and notably, the females are much more affected. So now the rate of female is 4 times more likely to be depressed than the males during the freshman year. Is it genetic? And before the pandemic, there was some signal that polygenic risk score was important, but that completely disappeared during the pandemic. A lot of people with low polygenic risk score for depression were actually highly affected. And by contrast, there are people who have very high genetic load who never experienced depression or anxiety during the pandemic. What is very highly predicted is a score that we developed called the affect score, that it turns out is we can get it from a variety of questionnaires. We used machine learning to develop it, and is highly predictive before the pandemic, the first 2 years of the pandemic, and continues to be extremely highly predictive. In other words, a kid can come in, and by the first day, we can predict with very high clarity whether they're very vulnerable or not to the stress of being a freshman and beyond. And it captures also chronicity and how long it sticks into the future. So I think we can show that we can use tools to predict susceptibility. And the question is, I showed you that resilience does not have to be genetic. And the question is, can it be learned? And we're in the midst of trying strategies to use cognitive behavioral therapy app broadly to see if we can target kids who are highly susceptible and see if we can change their risk. This is a collaboration with colleagues at Weill Cornell, Faith Gunning, and Francis Lee. But more importantly, I just want to emphasize that resilience is not just simply the absence of vulnerability. It's not just genetic. It can be learned and acquired, and our job is to figure out what happens in the brain and what we can do to induce that. My more general message is this. This is a paper that I co-wrote with Eric Nestler about what science can do about the mental health crisis. And I think it resonates with everything you all have been saying, that this is something we can rise to the occasion, we can do a lot. Understanding the biology of stress and how to induce resilience is one of them. Focusing on prevention, I think, is really important because there is bad plasticity. And of course, using knowledge to develop better treatments. Thank you.
Okay, some disclosures here, but I'll just get moving. So I started graduate school almost exactly 20 years ago. I was coming off a brief career as a physician assistant in school at Yale New Haven Hospital. And I was doing at that time was doing the early days of fMRI, and I was just completely fascinated by how you could put someone in a scanner and look at how their brain's functioning without actually even touching it. So I immediately went back to school. I said, I need to go do a neuroscience degree. Went to Washington University, and it was a very good time because there's lots of new discoveries, new techniques, like what's called resting state functional connectivity MRI, which allowed us to make some new discoveries that today essentially kind of slip off the tongue. We were identifying new systems that you can show up here, like the salience network, default network, frontoparietal systems, very large systems where your 100 billion neurons are kind of coalescing into large-scale systems from 10,000 foot up that are involved with interoception, rumination, mentalizing, attention, and anxiety, and things of this nature. Now, at the same time as this was going on, as we started learning more about these systems, we started collecting more and more data, big data, across the individuals. One of these large studies, which wasn't probably the first that kicked off, but it was shortly after, was called the Adolescent Brain and Child Development Study, where you can collect these types of data on thousands and thousands of kids. In this case, 12,000 kids, starting at 9, 10 years old, that we followed for the next 10 years. And now we're almost in year 10 or so we've been following for a very long time. And off that, other studies started, so other big data studies like the Healthy Brain and Child Development Study, which is the same thing, but starting at the neonatal period and following until the year 10. And then another aspect of big data was what if instead of collecting thousands and thousands of individuals of kids, you start to collect a lot of data, precision information just in one individual. That was kicked off by on the bottom left, one of my colleagues by the name of Russ Poldrack, mad scientist, who put himself in the MRI scanner twice a week for 10 months, collecting 14 hours of data on just one person. And these two sets of types of data set us off in some new space. I'm going to cut to the chase here a little bit and talk about some new discoveries, 2024, that are kind of changing the way we think about the brain. And they come in two different forms. One is large N, population-style studies from like ABCD, and small n, small individual personalized information just on one participant. So if people are interested in this, there's a couple of good papers here that you can see that describe a little bit where the field's changing. So let's talk about large N, population studies. I'm just going to give you two use cases of these two different styles, so it just gives you an idea and some fodder of why this kind of work is so incredibly important. All right, so this is the distribution of all the different sites across the U.S. that are collecting data for, this is actually the HBCD study, but a similar map exists for ABCD2, okay? This provided some very useful information for us because now instead of doing studies like I do in my lab where I get a bunch of people from my local city, I measure their brain, and I measure a bunch of cognitive measures, I correlate them so I can determine which parts of the brain are related to cognition, right? Well, now we have a bunch of other information, like, for example, this is the Child Opportunity Index, which measures, which essentially the combination of socioeconomics, environment, things of that nature of our youth. Now, Scott Merrick and colleagues and one of my close friends and colleagues, Nico Dozenbach, started looking at doing my experiment that I did in my lab, except for instead of doing it on one measure of cognition, doing it at 649 measures across this big ABCD study to determine, okay, which aspects of the brain are most highly correlated with what parts of behavior. And the gist in the bottom left is what you can kind of see there in the blue, as what soaks up almost all the variance in our behavior is socioeconomics and where we live, okay? It's not cognition, it's not mental health. The biggest factor that relates to our brain variation is where we come from, okay? Now, it's not just that. Of course, it's related to things, and Vivek Murthy set me up really nicely, is that if we look at the data that we can collect, that it's things like sleep, screen time, that seems to be driving a lot of these effects related to socioeconomics. It's just a fascinating result, has huge implications for how we conduct our research, policy, things of that nature, that will last for a long time. Just my first anecdote, my use case 1. Now here's my use case 2. Now how about the high SNR? So in this case, I'm going to be just showing you, this is 200 participants from the ABCD study, just showing you that these general networks that I've talked about earlier are in everybody, but they're highly personalized. They're very specific from person to person, okay? that variability from person to person actually matters. It means something. And I'm trying to, knowing who my next speakers were, I'm trying to set them up a little bit. A paper that just came out just 3 weeks ago by Chuck Lynch and Connor Liston showed that the size of that salience network, that first network I showed you in that first slide, is directly related to depression. Okay, in this case, major depression, where these larger salient systems, these markers of larger salient systems, seems to be highly related to depression. On the top, there's 3 examples of those cases. On bottom, and there in C, shows you initial data set and how this phenomenon is highly replicated. And on the bottom right, what it shows is that even before the onset of symptoms, and this is the case in the adolescent brain, that this is a risk factor for developing major depression. Well, while this study was going on, I was in the background working with one of my colleagues, Ziad Nahas, and several others at the University of Minnesota, where we were identifying the exact same phenomena in cases that were about to go to do cortical neuromodulation for depression. And we immediately identified, oh my gosh, this salient system is massive, in this case of a treatment-resistant depression patient, a patient who has 30-year history of depression, multiple hospitalizations, 3 rounds of ECT, suffering cognitive decline, tried to commit suicide 3 times, only survived once because the gun jammed, the other time because the police officer found him before he asphyxiated. There's massive, massive problems in this patient for the majority of his adult life. Now, if you have a large, expansive salience network, that also means that it's encroaching on other systems that exist too. In this case, there's other systems that I showed you that we discovered 20 years ago, right? The default network, frontoparietal network, this is what's called the attention mode network, that have all these kind of different functions were also atypical in this patient because of this massive salience. Now, because we can collect these massive amounts of data and personalize the specific systems, that allows us the opportunity to actually, in these cases of cortical neuromodulation, to target the specific networks, which I'm showing you all here on the left. The long story short, by knowing the function of these systems, by being able to target the specific systems in the specific patient, we're able to reduce, within 7 weeks, his suicidal ideations were completely gone. Within 6 months, the symptoms were 50% down. And within 9 months, he's in complete remission. I saw him 3 weeks ago. It's 21 months where he's been in remission, the longest period of his entire adult life of wellness. It's fascinating. But not only that, but also targeting these systems showed something else, which may be related to symptom decreasing, but also showed, which is something fascinating to me, that his cognition, even though he started off at average or above average, actually went up even higher after some of this treatment. It's an amazing case, but just gives you an idea of where we come. So I'm just going to end there and just say it's a new age. You know, we have these large samples like ABCD and HBCD that are changing the landscape of how we view factors that affect brain development in youth. We have these new techniques of precision imaging that allow us to monitor efficacy of interventions, targeting for neuromodulation, characterizing detailed aspects of physiology. And now that we understand, after 20 years, the specific parameters of how to do this well, it's a major advance, and which I call we have liftoff. So I'm there. Thank you.
All right, so we're going to switch gears again, and I'm going to be talking about medications. As you probably know, we have a large number of medications for the treatment of depression, a large number of medications for the treatment, antipsychotic medications for the treatment of psychosis. And despite having this enormous range of drugs to try, depression and psychosis, schizophrenia remain among the most disabling medical conditions that we have in all of medicine. And so that story could be very bleak, and we know that part of the issue, as we heard about this morning, are the difficulty that people have in accessing effective treatments. But a part that rests in neuroscience is the limitations of the effectiveness of our medications for treating the symptoms of these disorders. And so what I'm going to be talking about in my 6 minutes and 56 seconds is some of the advances that are making that are transforming the way we're thinking about the pharmacotherapy of psychiatric disorders, keying off of some work related to a drug called ketamine and esketamine. Esketamine was the first mechanistically novel antidepressant approved for the treatment of depression in 50 years. Following that, in 2019, brexanolone was the first antidepressant approved for postpartum depression. And just a few weeks ago, the first antipsychotic that doesn't block the dopamine D2 receptor was approved by the FDA. So neuroscience is now transforming what the future of pharmacotherapy is going to look like. So we'll talk a little bit about that. So why is ketamine or esketamine so different from other medications? The first quality is the rapidity of its action. With the standard Prozac-like antidepressant medications, you see clinical improvement over several weeks. It may take several months for those people who are going to respond to remit from their depression. Ketamine can produce improvement within hours. It can produce remission within a day, often with the first dose. If you have treatment-resistant symptoms, which is a very common, perhaps half or more of patients with depression, the chances of responding to one of these standard medications is very low, and double or even more the chance of responding if you get esketamine. Esketamine right now seems to be the most effective medication that we have for treatment-resistant symptoms of depression, and comparable in effectiveness to what had been the gold standard, which was electroconvulsive therapy. Something that people don't talk much about, but if you have treatment-resistant symptoms of depression and you get treated with an antidepressant and you're fortunate enough to respond, the chances of relapse over the subsequent year are very high, over 50%. And that risk is also halved or reduced even further when esketamine is used as an antidepressant. A surprising issue, because one of the distinctive features of ketamine and esketamine is their evocation of dissociative-related sensory changes, that people were concerned that there would be tolerability issues. But when you look in the large-scale clinical trials, and where esketamine is compared to, say, an antipsychotic medication, you actually see more people discontinuing the antipsychotic medication due to side effects than esketamine, because with esketamine, the side effects are limited to intermittent sessions, whereas if you're taking a medication every day, you have to live with those side effects. And then the long-term effect of these new treatments, illustrated by esketamine, can be profound. So there is 42-month data that were analyzed by Johnson & Johnson, and they compared it to a historical large-scale data set. It was about over 1,000 people treated with esketamine. And over those almost 4 years of treatment, they showed a 10-fold reduction in the odds ratio of suicide attempt, a 5-fold reduction in the odds ratio for death by suicide, and perhaps for this audience, most importantly, a 3-fold reduction in all-cause mortality relative to standard treatment. What I mean there is that, as you've heard, depression worsens the outcome from nearly every medical condition, and standard antidepressants help there, but these new medications have an even broader effect. Don't think we have time for this, so I'm just going to skip to the next slide, which is one of the important things to appreciate about these new medications is that they're emerging from new ideas about the biology of depression. So for the first 20 or so years 30 years of antidepressants, people thought that the effectiveness of serotonin and norepinephrine reuptake inhibitors meant that depression was an illness of the serotonin and norepinephrine neurons in the brain. And what led to the discovery of the antidepressant effects of ketamine was a shift in focus to think about the intrinsic signaling mechanisms of the cortex and limbic system, which use glutamate and GABA as the primary transmitters, leading to the testing of ketamine as a glutamate receptor blocker. And in turn, it is now evident that ketamine can restore function, structure, and function in the glutamate, the main, most common form of signaling in the brain. It can restore synaptic density, it can restore synaptic effectiveness, and it can reduce the detrimental effect of neuroinflammation, which is a part of the biology of depression on the brain. So I thought I would just show you a little bit about what I mean by this structural effect of these rapid-acting antidepressants. What you see on the left, I hope for you, is the farthest left is a dendrite, an input to a nerve cell, and you see that there are a certain number of little blebs off that dendrite. We call those blebs dendritic spines, and they're the places that glutamate neurons make synapses with each other. What you see in the stressed animal 24 hours after a single dose of ketamine is a remarkable proliferation, really from the work of Connor Liston, we know regrowth of dendritic spines. This is a figure from a paper of my late colleague, Ron Duman. What you see on the right side is data from a positron emission tomography study where we can label glutamate synapses in the brain using a tracer for a protein called SV2A. And what you see is that the degree of improvement, in other words, I can't read you'll have to take my word for it, the degree of improvement in depression, which is going to the right on the x-axis for me, is associated, the level of the increase in synapses, which is going on the right in the x-axis, is associated with the degree of improvement, in other words, a lowering of the depression score. So we can measure these synapses. They're low in patients with depression, and within 24 hours of a single dose of ketamine, we have some early evidence that these number of synaptic connections in the brain are restored. Well, what about psychedelics? Hopefully by now you've heard somewhere about the idea that psychedelic drugs and MDMA may be another form of novel antidepressants. Some people describe the response to drugs like psilocybin as among the most important experiences of their lives. Like with ketamine and S-ketamine, psilocybin appears to produce a rapid and robust and sustained antidepressant effects that periodically with repeated dosing can be sustained. While the initial target in the brain of psychedelic drugs like psilocybin is different than the initial target for ketamine, they have some convergent effects on the brain, engaging certain common signaling mechanisms, stimulating glutamate release, and triggering the regrowth of these dendritic spines. One of the challenges with psychedelic drugs is that the subjective effects can be overwhelming, and they can even trigger suicidal thoughts in people as a consequence of being overwhelmed by the content that comes up. And so this is triggering innovation in neuroscience as various groups are pursuing drugs that may engage some of the same signaling mechanisms as the psychedelic drugs without producing hallucinations. But this is an extremely fertile moment in psychiatric neuroscience, particularly around medications, and a wide variety of mechanisms are being implicated with novel treatment mechanisms for depression and, as I mentioned, for schizophrenia as well, with the FDA approval of KarXT Cobenfy. So I thought I would close by highlighting two great challenges. First, the enormous heterogeneity of patients with our psychiatric disorders and the need to be able to biologically and clinically subtype these people. And the second, the need to translate from the complex genomics that Dr. State talked about to novel therapeutics. With that, I will pass it on. Thank you.
So thank you, Kaf, for inviting me and for reading my bio. I'm going to kind of go backwards and actually go back to the future in some ways, because I want to tell you a bit about basically taking the knowledge from neuroscience experiments that you can do in people and actually use it for a therapeutic, and how we can build on the technology advances over the last 20 years. I'm going to talk, and I think it kind of speaks to the fact that all of this is about community. The teams that do this work are totally dependent on a team of expert clinicians, not just a psychiatrist, but a neurosurgeon, a behavioral health expert, someone who can do therapy, but then in concert with imaging experts, computational experts, neuroscience experts, modeling experts, and actually now leveraging everything about technology from, we'll say, rodents to AI to actually accomplish this work. I think I want to kind of reiterate, I'm going to use depression as the example, but that the need is obvious. All day we've heard about this. And I want to build a little bit, not as a poke, but the idea that even with these advances, with these novel pharmacologic therapeutics, we need to get people well and we need to keep them well. And if people don't stay well, then we have all the problems and we just kind of put bricks into the wall and they fall out the other side and we keep going back to where we started. So what do we do? What is our strategy if we fail ketamine and ECT? Because they have had remarkable progress. How can we deconstruct this and think about this? Well, I'm a neurologist by training, and basically it comes down to the wiring diagram of the brain. When I trained, and it just makes me realize how old I am, Damien, is 20 years ago is when we implanted our first DBS patient, and it was 15 years of work thinking about the neurology of depression without fMRI, with basic tools of imaging, so that everything I'm going to talk about for the next 5 and 45 seconds is about how knowledge about wiring diagrams of the brain and then the chemistry within those wiring diagrams has informed not just these advances in treatment of depression, but other disorders. And this is really all off the backs of thinking about movement disorder, Parkinson's, where the late Mayland DeLong, member of our great community, died a few months ago, but that he and Benabid really were the first to pioneer DBS for movement disorder. It didn't come out of the air. It came from understanding the wiring diagram, the physiology, the chemistry, the degeneration, the basic biology of Parkinson's disease, and then leveraging, by being brave and clever, technology to go into the brain when pharmacology had failed. So again, it's counterintuitive that if we understand the biology, we can go into the brain. But actually, if we can define the wiring diagrams, which we have for OCD, for addiction, for seizures, for movement disorder, and also in depression, then we can ask the question of where do you stimulate? What should happen? How are you going to deliver it? And who should deliver it to? And that kind of template is really where the field of focal neuromodulation brain-computer interfaces now are. Now, the story of depression, because it's the only story I really have to tell, is that you can go back to a 1994 image of a healthy person and a depressed person with a PET scan. And you can see that the frontal lobe, it's marked, is abnormally low. And that was the best replicated finding. It's been expanded by thinking about abnormalities in the default mode network, salience network. But as you started to look at people as they weren't responding, you start to see that it's a contagion, that other areas also come into play. And an area that became kind of couldn't look away was this area in the subcallosal cingulate. It talks to both the salience network and the default mode network, but it's not a member of either. And what we could start to do was build these kind of line and dot wiring diagrams and actually leveraging the technology that had been done for Parkinson's, knowing that you could precisely place a wire into the brain, apply a duty cycle of focal low, high-frequency stimulation, and block the activity where you put it. Well, we wanted to block area 25 and figured that if we affected that node, it would affect everything that Area 25 was connected to. You could see it on an MRI. You could plan with the surgeon to put these small wires with millimeter precision deep in the brain into that spot, hook it up into a battery pack, turn it on, and you get actually an effect on the operating room table of the intense psychic pain, and the inability to move away from it resolves immediately, and nobody actually has an altered state. But you're not done, because even though there's this first effect, you are not well. And over weeks with continued stimulation, people get better and stay better. And we have patients that are well with the device now 19 years. So again, the other people have had different logic, different areas of the brain. Meta-analyses on these small studies are positive. This is still not available as the general public. But what we've been able to do is to learn that despite the complexity of the brain, as Damien has shown, it's not a spot, it's a network. And if you take people who have recovered, you can start to reverse engineer in small samples to realize it's a convergence of a set of connections to a set of these networks. And that one can, once one knows where in this morass you are, you can actually put the electrode in individual patients and match what you could get in the group. What's happened is, despite this precision and this projection of how people recover over time, recovery isn't linear. Some can have the same score and actually be having a very different experience. And the experience is expressed to their therapist, their psychiatrist, and the psychiatrists are trying to decide every time they see a patient, are you needing to have an adjustment, or are you needing to change something in your environment? And how do we look at that? Well, what we do is we need readouts, and this is where, again, technology has been essential. The device companies have built in tracker signals, sensing signals. It isn't just in the lab or in the hospital. You can actually track the activity in the brain on the device where you're stimulating and basically have a beacon as what's happening. You can put people back in scanners. You can start to actually use the patient as a collaborator. Let them explain what's going on. We have patients recording from their brain, doing video diaries, and now in the last few years with the explosion of machine learning, natural language processing models, we can actually dissect how people talk to themselves about their experiences at home and actually use that as a tracker. And we can get fancy, we can build things in the lab, we can utilize other machine learning tools to look at other aspects of behavior and have some fun while we're doing this work. But just to kind of say, what we've discovered is because we know we can get people well, we can actually have these tools that we can track over time. We can actually just listen to what the brain is doing and use machine learning at the end when they're well compared to the beginning when they're just different, and actually devise tracker signals that can allow us to watch patients and actually know when they might be getting into trouble before they do. And we're starting to learn that maybe we can do that without the tracker signal in the brain by these kinds of information that patients can do. So this is like the most exciting thing, and I want to say it because it goes back to the idea of plasticity, the idea of knowing the network, knowing what's wrong in the brain, knowing how it might have gotten there, because we're dealing with patients that are at the end of the road, the absolute end of the road. And so what we've learned is as people get well, people get better at different rates. And the rate at which they get well is dependent on how damaged the connector cables are in the brain, how damaged the white matter is that connects these collections of neurons to each other across white matter cables, that we have scans that we can monitor that. And now that the tools in their brain, we can actually put them safely in the scanner, we can actually see that the areas that are damaged over time are actually repairing. So while John talked about plasticity and synaptic plasticity at the neuron, we're actually rebuilding the broken cables that connect these systems together. And, well, it was just a scan, and people go, well, that's not really real, that's an inference. We can take animal models, and this in macaques who have very similar brains to ours, and we can stimulate, even in healthy macaques, and we can see how the brain remodels with a period of stimulation, and actually can look at the electron microscopy and see that we remodel the myelin. We reconnect the broken brain. So I just want to end with the idea that now we've reverse engineered with an idea, have deconstructed what the signal is. We have a way to know precisely how to personalize in individuals. Game isn't over, because as we repair the brain, now we need rehab. And I think I need to substitute this slide with one of a broken leg. Nobody says to someone who has a skiing accident and sits and has a pinned leg, sits in traction, then goes to a cast, then goes through months of graduated rehab, that, hey, surgeon, fix your leg. The rehab is up to you. Maybe you can get it. Maybe you're lucky enough to be on the Olympic team. Patients are getting better over time. Where they are in terms of their acquired resilience, probably based on remodeling their brain, is due to the fact that they're practicing, they're seeing therapists, they're actually getting activity-dependent plasticity. And we get back to where we're actually kind of all in this together. People always say, no, you're just dealing with a small sample of patients, this doesn't scale. If we understand in our most ill patients what is going wrong, and we can move it back, we can return it to other and use combinatorial treatments, including what we can do in the community to actually help these people retrain their brain.
So I thought we'd have a brief discussion, and this has been mentioned by several of the speakers. We've had themes around large data sets and what they've allowed us to do, large genomic data sets and finding convergence, stimulating the brain, tracking folks across space in their local environments. And I'd love to explore this idea of how we process and manage big data, particularly through the theme of artificial intelligence. So in the last few years, all of us have been taken by storm from ChatGPT coming out and seeing what it can do. But even as of recently in the area of the health sciences, a few weeks ago, the Nobel Prize in Physics was awarded for artificial intelligence and machine learning, and the Nobel Prize in Chemistry was using artificial intelligence and machine learning to model proteins. So I'm really curious about how managing, processing these big data sets and artificial intelligence plays a role in the work that you all are doing.
We'll go down the...
We can jump on in.
It's free discussion. So I had a brief example, but we've been engaged in this process, psychiatric cell mapping initiative, for now 7-8 years. And the real challenge is that what we're doing is we're taking autism genes, cloning them, doing affinity purification, mass spectroscopy. And the challenge there is that you do generate this enormous amount of data, and actually there's something called the crapome, which is what previously people did to try to figure out what was real and what was not real by subtracting out the crapome. So we were struggling with that for many years, and then we had the tremendous good fortune of doing the experiment at the right time. It's always better to be lucky than to be good. And so we had AlphaFold, right? And so what AlphaFold enabled us to do was to take, as I said, about 30,000 potential interactions, with a small fraction of those actually being kind of the direct interaction between autism-related molecules that we wanted to look at, and sort through all of that with incredible rapidity and with great both specificity and sensitivity. And then to take the step beyond that, which is where this is moving now, not just to see how the proteins are fitting together, but actually to get atomic level resolution on the changes in the human genome that are leading to disease and what that does to the structure of those molecules. So that it kind of threw open the doors on a data set by allowing us to dig through a massive amount of noise to get its signal in a very rapid way. We're still overwhelmed with the amount of that signal, but it really did transform and sort of, you can see this setting the stage for really a quantum leap in being able to think about the scale of proteomic data sets that now will no longer be overwhelming because of the ability to leverage AlphaFold.
Yeah, so I've thought about this a lot because I think a lot about individual differences. Your point about how each brain is unique, even though as neuroscientists we're looking for commonalities, but everyone is different. And I think the opportunity to recognize both those commonalities and differences and translate that into something meaningful about maintaining our own mental health and mental health of our kids, our community, actually can profit immensely from the ability to integrate data in possibly as time goes on, based on both evidence, biological evidence, behavioral evidence, response to certain treatment strategies, whether it's an app or game or whatever, and do that in an informed way that is personalized. Truly not precision, actually potentially personalized. So what I do for myself is different from what I do for you or what I do for you. I see this potential, and I feel it's especially important in terms of scaling when we're talking about disorders that are not rare but are common, and where we can't do that individually. We have to put it in the hands of something, the person, how you're sleeping, how you're walking, all of that. I think that is the hope, and that is why this idea that there is hope, there is room to move, we're not trapped. We just need to pull together the right players in the right way to help the person in the right moment before things get out of hand and they need to see my colleagues. That's where I want to get to them, when they're young and before the problem gets huge, and I think AI can help with that.
Yeah, you mentioned your ability to predict the freshmen coming in, which ones would do well and which ones won't. How early do you think we could get with those sort of prediction frameworks? Would it work in high school freshmen? Would it work in earlier grades?
Yeah, I mean, Dr. Murthy said this morning, if you listen to people long enough, they'll tell you what is wrong with them. And these kids actually, with a bunch of specific questionnaires and a little bit of biological data, actually are telling us what is wrong with them. These are psychological disorders in the end, and people have some insights, some better than others. We haven't gone back enough in time to know if a 10-year-old can tell you, but definitely an 18-year-old is telling you where they're at. And I think we should not throw that information away in our rush to be very technical and very biological. The information is there, and it's actually continuous. It's not that the depressed people are one way, the not depressed people. There are people who are slightly there, closer to the edge, even closer to the edge. That information is there. We can get it and we can use it, I think, and we can see where they veer off closer to something catastrophic or how we can pull them back.
Yeah, I was gonna say that it's the opportunity to your question is like immensely high, and being kind of a lead on some of these large data sets, what I'm certainly learning is that there's no way that any given lab, any given person can really recognize or realize the potential of a lot of these data. We need to be working with informatics. Sometimes I feel like I'm the informaticist because I'm doing more informatics sometimes than neuroscience now with these big data sets where, as Steve Hyman used to say, you got to break out of our cognitive boxes and get into other spaces and understand to really harness the power of all these data. It also requires a big culture shift. We're so used to just doing things the way that we do it, but to really build on the capabilities of all this data that's being collected, it needs to be standardized, made more shareable, generalizable. That's actually kind of hard to do, and it requires huge shifts in the way that we do things every day. It's like you're changing culture, right? And the last part is that, and it's related, is the quality, right? None of these things work if the data is bad, And so, how do you, the only way then to collate, harmonize, and then QC these data is we're kind of doing it as groups, as collaborations, as big teams. So it is a big change. I think the power is there. We can feel it, you can taste it, in the world every day. You see it arising. But in our space, there's just a little bit of extra. It's a new world of getting into informatics and other spaces that we just typically aren't used to.
So I think I'll take a clinician's perspective on your question, and come back to the enormous challenge of heterogeneity in psychiatric disorders, which is a huge problem. And so we have a diagnostic manual I don't think any other branch of medicine has a manual that defines the diagnoses. But in psychiatry, we have a diagnostic manual. And in the diagnosis of major depression, you can meet that diagnosis in 227 different ways, often with symptoms that are opposing each other. In other words, too much sleep, too little sleep, eating too much, eating too little, being agitated, having no energy, and being very upset and being unreactive. And one of the challenges that we face when we try to choose treatment for a patient is that we don't know how all of these individual variables predict the response to the treatments that we're prescribing. And so one area that seems to be very enormously promising is to use machine learning to process all these variables at the same time. And so a graduate student that I supervise, Adam Chekrud, did a series of studies looking at the very item-level data from enormous clinical trials and showed that you could do, using these machine learning approaches in ways that were replicable across clinical trials, predict about twice as well as the average clinician how well a person was going to respond to medication. And so, and not only that, but the predictive models were defined by mechanism. So they did a better job in predicting antidepressant treatments that were within mechanistic class than in predicting response that were a distinct mechanistic class. So I think this idea that AI will help us parse heterogeneity, which is one of the really great challenges that we face, I think is really a very promising approach.
So to add to my colleagues, I mean, machine learning and AI kind of set us free. And I think part of it is the culture shift of, as scientists, you have a hypothesis, you test it, and you have a null hypothesis. It's very slow, and it turns out it's biased in a very different kind of way. And so to combine a hypothesis, which ensures you have good data going in, with then an almost agnostic approach with machine learning, has really changed the way even our work has evolved. And I think that you let the machine tell you that actually, despite what you know is absolute heterogeneity in even these most severe patients, everybody has the same signal that tracks that you would not have predicted, but the data reveals. And where the variance is actually going back to what Huda says, and I was writing down, I need to get the predictor for who in college, but actually machine learning has told us through the patient's own diaries that even though they all get well, we can actually now predict the ones that actually have trouble using being well. And I'm now getting to where, in a heretical way, I don't think depression is heterogeneous. I think people are all different, and their environment is different. And we're back to what Huda is saying, is that at risk, I'm taking care of and trying to reverse the thing that has happened to the vulnerable brain. But once it's repaired and stable, you're back to how to train the person in the temperament they have and the environment that they live in. That introduces a lot of the variability that I think even Damien is seeing when you sit and have Russ Poldrack, scan himself. I'm sure every day is not the same twice a day. So I think the machine learning, I do want to bring up something we haven't talked about. The biggest source is what patients say themselves, their willingness to share. The fact that we hear that people go on chatbots and are using a chatbot as their source of therapy because they don't have access. That's a problem. Because as a chatbot, as we heard today, can feed you, maybe it's just better to kill yourself, that we need to actually also train these devices if we're going to use them in a responsible way. I think the other thing that we have to deal with is, in the culture shift, I cannot share the videos that my patients have generously recorded for my team to use. I'm not able to share them with other experts who might be able to apply these deep learning algorithms in a clever way, because they are patient health information. And so again, if we're going to have that, you can be spied on in the culture by a machine with your kid in their bedroom at night, that I should be able, with the patient's permission, to share their data that can help them to be better and to develop new treatments with others that have a like-minded mission. So I think we need more than the cultural shift of psychiatry. We need a cultural shift of science and society.
Well, thank you for those thoughts. We'd now like to get you all involved in the conversation. If anyone has a question, please feel free to step to one of the microphones. And if you're unable to make it to the microphone, just put your hand up and someone will bring a microphone around to you. Yes.
Thank you for a superb set of presentations, very exciting. I'm Linda Freed, I'm at Columbia, and I wanted to maybe build off Dr. Akil's statement that there is potential for prevention at scale to build resiliency to depression. And I'd be interested in hearing from each of you about where you think prevention could be effective at scale in communities, in families, at a policy level even.
Well, in the interest of the 13 minutes we have left, well, you'll hear probably from two of us, but we'll all be around later afterwards. So please.
Well, I'm not a policy expert by any stretch. And so, but I feel that it is what I tried to say earlier. First, you have to predict. And the other is what we talked about, is that we have the tools. So theoretically, just with a little bit more time, it should be possible to identify people at risk and take the knowledge that we have, combine, and begin to figure out, at least theoretically, the pathways to doing better. There are lots of therapies that are turning into apps, for example. They don't fit everybody, but you can figure out how to, match better. And you can then tell systems, whether it's my University of Michigan, which has been wonderfully supportive of my effort, whether it's universities or workplaces or states or schools. There have been questions about schools. We need to take it seriously. But as scientists, we need to begin to offer workable solutions that can be scaled and then communicate with partners, whether local communities, government systems, and do real-life tests for what is workable, whether it's rehab or prevention, and see how we need some success stories before it can be recommended. But the last thing we should do is, it's too hard, it's too complicated, we keep needing to do more science, and meanwhile people can suffer. That's what I don't want. So that's why I'm pushing the message that science can do something now if we partner correctly. It's not exactly an answer, but it is the best I can do.
I'm just going to take that on a little bit. Even at the first episode of depression, with imaging, you can actually get very good separation in the same circuit of who will get better on drug and fail therapy, who will get better on therapy but fail drug. And I know when we first published this 10 years ago, that it was met with, well, it's not like we're going to scan people to decide how to treat them. And I don't want to waste more time by the idea of say that to a cardiologist before they put in a stent, or someone treating with cancer before they decide on a chemotherapy. But it is a reality. But with machine learning, even at that level, we can take the scans and we can start to say, is there's an easier form where the information correlates. So we use the neuroscience and we move backwards to something that may scale. We don't abandon the neuroscience. We use it as a bridge to something else. I think the same thing goes through by identifying nodes or systems or cells or chemical systems where there's failure, and that, again, with whatever tool we have that isn't scalable, we look for ways in which we can scale it. And I think that's how we'll really bridge these. I mean, we can't keep up scientifically with what's happening in the private sector to use these technologies, but we can figure out and cherry pick when they can actually serve us, and that's where we can also, I think, form collaborations to get to the kind of question you say. So it could be that we're screening kids early or in high school. It's not intrusive. It doesn't require the resources. That means everybody can't do it, but it's evidence-based. It's not wishful thinking.
Thank you very much.
I'm just going to add one thing in this, and it goes back to my idea about how important it is to collaborate outside of our spaces. We can make discoveries like prediction of outcomes and things like that as scientists. We're not very good at like handing the baton off to the policymaker, handing the baton off to the industry partner who can actually take that and do something with it. It's like a huge hole in like our thinking often, that we need, like that's a huge part of it. We can discover stuff, but it doesn't mean anything until you applied. And it only gets applied if we hand it off in efficient ways to people who can actually do that.
Roger Johns from Johns Hopkins. Hello, Huda. Huda got me started 50 years ago in her lab as a gopher undergraduate. The beauty of what you guys have presented today is bringing the complexity of pathway biology to neuroscience and to major neurological disease processes. I think adenocarcinoma is one of the first that did this in looking at about 350 different pathways by which you can result in the same outcome of uncontrolled growth in adenocarcinoma. Beauty here, I think, is that you're doing this from different perspectives. And the treatments could be tracked, perhaps, these are neural pathways. The neural pathways, the signaling process, is a way of tracking which of these complex molecular pathways are interacting. And then you can stimulate it more specifically. But together, you guys all have an amazing laboratory that could, I think, come together in these processes. Anyhow, thank you. Wonderful.
Thank you.
Keith Martin, Consortium of Universities for Global Health. Thank you very much for your presentations. What you said was really profound from a policymaker's perspective. And I just plead to you that I hope that your presentations can be shared with state health officials and educators. Because what you said can have really profound implications for children across this country and their future trajectories in their lives. But my question to you is, I think you made a comment about the opportunity for engagement is at a high school level. But if neuroplasticity is greatest in the first 5 years of life, can you give a sense of what can be done from the prenatal stage through the first 5 years of life that can dramatically affect the temperament and the adaptability of the individual in terms of their ability to affect and deal with stresses later on? Thanks.
It's a great
Start.
Okay, it's a great one. So at my, I've been dealing with this actually quite a bit. The institute that I run, the Masonic Institute for Developing Brain, one of the key periods is this first 1,000 days, the idea of in these very early years. And I think there are some very basic things that can be done. I mean, most of our investment in our kids starts at school age. I mean, most of our policy investment starts then when things like just nutrition, access to early care, very basic elements of like interactions with our kids at these very early ages is probably the place where you can easily start. It's one of the things that we're working on actually with our Governor Tim Walz, that some of you guys might know, in Minnesota right now. So there's some very basic things like that. And scientifically, of course, we're learning a lot about the prenatal environment and how that affects long-term trajectories in our youth. But I think there's much low-hanging fruit because there's just lots of room to invest in these early stages of our kids.
So it's absolutely true that the earlier the better. But my perspective as an old person, very old person, is that it's possible to maintain neuroplasticity for many decades. And I think there is an art to it. I think it is like knowing how to inherit some money and keep it going and multiply it instead of spending it down. So your earning power may be higher at certain times on others, but you have the art of creating more. And so I really think that while there are windows of time where plasticity is, highly available and you can use it, I don't think it ends. And I think that is fantastic news. And by plasticity, I mean both good and bad plasticity. Learning the right things and learning how to cope, and learning the wrong things and getting stuck into a rut and bad behavior. Not bad behavior, but bad coping. And so this idea of experience, the reason I wanted to focus on experience is you could see that you react one way and then you build on it, Can you deviate from that? Adolescence, I see as a time where the gonadal hormones kind of reopen the chromatin, and there is a lot of rewiring going on, and it's a fantastic time. And in our very genetic animal models, we can really flip the phenotypes during adolescence in a way that's much harder to do in adulthood. And we can use it through experience, we can use it with growth factors, so artificially as well as environmentally. So I just want to kind of counter the idea that it is just one-time window. But the earlier, the better. And the more we put this idea that neural remodeling is an ongoing process that would work into old age, I think the better it is for all of us.
I think the fact that...
All the old people love me. Thank you.
Message for this class.
Exactly. This is the right audience.
So let me just check to see if there are any live audience questions, online audience questions before I take out. Okay.
Albert Reese, Maryland. First of all, thank you very much for very, very spirited discussion. This is very enlightening. I'm a physician scientist, but in maternal fetal medicine. So my question is, do you see risk, since the brain there's plasticity, then obviously the sooner you can make the diagnosis, in theory, the better you can make an intervention that's effective, in theory. So my questions are as follows. Number one, are there sufficient risk factors that you can at least begin to do early screening? And number two, whether or not there are in utero opportunities for diagnostic purposes so you can truly make reversal in utero.
That's you, Matt.
No, baby.
Child psychiatrist. That's a very broad question. I think so. Clearly, there are opportunities. I think to leverage the kind of things that we're looking at to think both about very early identification of risk factors and early identification of phenotypes. I'm a child psychiatrist, so by definition, the things that we're looking at emerge early, and when we're talking about autism, it emerges in the first 3 years of life. And there are already a variety of tools, some leveraging AI, but leveraging a variety of sort of new quantitative behavioral phenotyping measures to try to see whether or not we can move back from about 3 years of age to get reliable diagnoses now to about 18 months of age. And I think there are clear opportunities for the broad group in autism to be identified in the first year of life. There's suggestion that there are early indicators that may be captured by new methods. And I think, we get, whether it's ADHD or early onset depression, et cetera, I think it certainly is reasonable to assess that we can, as I said, use the kinds of things that people are learning about and that you've heard about all day today quite early in development. With regard to in utero, because of the kind of, as I mentioned, the distinctive genetic architecture of some of the more severe early neurodevelopmental disorders, those diagnoses can potentially be made in utero with enormous ethical challenges to thinking about how you would manage that, particularly because except in some very extreme cases, even when we see a mutation that has a very large effect and we know is reliably associated with autism, there's not a guarantee that that's going to be the phenotypic outcome, and that we see see such broad range of outcomes from single mutations that the kind of risk benefit of thinking about prenatal intervention in order to try to address and identify genetic abnormality, even one with very large risk, I think is fraught and presumably, I think, quite far away for the kinds of disorders that we're talking about.
Let me just say, it was a tremendous honor to be here with you all this afternoon as I close out. I love the question about taking this to the state houses. I was talking to my colleague Brandy earlier, who's one of the NAM Science and Policy Fellows, who's in the Colorado State House. And I think it really highlights the image that Huda put up, which at the bottom it was genes and cells, and at the top it was state houses, right? And I think it's by combining work, thinking about genomics all the way to brain imaging and thinking about how that's related to socioeconomic status, to thinking about the complexity of the brain and the wiring diagram and how to stimulate it, and also thinking about how we can advance pharmacological approaches that we will ultimately get to a place of health. It is not neuroscience or the rest, or loneliness, or thinking about early childhood education. It is neuroscience and those things. So thank you all for having us here this afternoon. It's been such a pleasure.